I'm Madam Joyce from Educare TZ. I'm teaching biology, and today I'll be with you to discuss our topic, which is gaseous exchange and respiration. And here we are going to deal mainly with our subtopic, which is the respiration. So as usual, we have to recall on where we end on the previous session. We end by discussing on the process, which is the glycolysis. Now you can see we have almost 3 stages, which is phosphorylation of glucose, lysis, and the oxidation by dehydrogenation. So my student, as I promised you in previous session, that in this session we are going to see the figure which now summarizes the whole process of the glycolysis. So if you don't kuelewa kule kwenye zile explanation the glycolysis ilikuwaje kuwaje, so now here my student, I hope you are going to understand, and you will talk, and you will have now something that you can explain by your own. So now you're saying that the formation of pyruvates and the glycolysis processing. And now let's see this figure now, which explain the process of glycolysis and the formation of the pyruvate, which is the end product of a glycolysis. So now let's start looking on our process, which is the glycolysis. And we're saying that the first stage, which is the glucose. My student, first, before starting discussing this figure, you have to know that the process which is the glycolysis, it is under control of enzyme. So each stage there must be the presence of enzyme, which now will catalyze that reaction, so that to form a certain product. So hapa kwenye first stage of glycolysis tumeona kuna presence of glucose. So in order for breaking down this glucose molecule to form the glucose 6-phosphate, we use the presence of ATP. We are using the ATP, the first stage. So but this reaction, as I have said, this reaction, they are controlled under the enzyme. And the enzyme which is responsible for converting the glucose to form glucose 6-phosphate, it is known as the hexokinase. And after formation of this glucose 6-phosphate, now this molecule now it will be isomerized to form the fructose 6-phosphate. So in order to be isomerized, meaning that this molecule and this molecule, they have the same molecular formula, but they have different structural formula. Na ndio maana tunaifanyia isomerization, so that hii molecule isiweze ku escape outside the cell. So now we are using the enzyme ambayo inaitwa phosphoglucose isomerase. And after formation of the glucose 6-phosphate, we have another enzyme, so that anaweza akatusaidia kwenye formation of the fructose 1,6-phosphate, meaning that in this reaction we tend to add a phosphate group. Na ndio maana hapa tunatumia hii phosphofructokinase. So huyu phosphofructokinase ndiye ambaye atasaidia addition of a phosphate group into a fructose 6-phosphate, so that we can form fructose 1,6 biphosphate. And after formation of the fructose 6-phosphate, we have another enzyme ambaye ndiye anaitwa aldolase. Huyu aldolase atasplit hii molecule ya fructose 1,6 biphosphate kwenda kupata 3 carbon sugars. And those 3 carbon sugars, they are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. That is our first product after lysis of the fructose 1,6 biphosphate. And we have seen that this molecule, which is the glyceraldehyde 3-phosphate, ndiyo ambayo itaendelea na ile glycolytic pathway. And also we have seen that in order for this molecule to undergo the glycolytic pathway, they must be rearranged, or they must be isomerized, because all of these molecules, they just have the same molecular formula, but they have different structural formula. So due to that, we have to convert this molecule, which is the dihydroxyacetone, to form the glyceraldehyde 3-phosphate. And this molecule, it is enhanced, or this reaction it is enhanced with this enzyme ambayo inaitwa triphosphate isomerase. So ataibadilisha hii molecule direct kwenda kupata hii glyceraldehyde 3 phosphate molecule. So now, after formation of the glyceraldehyde 3 phosphate under this enzyme, ndiye ambaye ataingia sasa kwenye ile glycolytic pathway, ambapo sasa hii glyceraldehyde 3 phosphate tumesema kwamba we tend to add the PI. We add the inorganic phosphate or phosphate group in this molecule. So after addition of a phosphate group, we are going to form 1,3 biphosphoglysylate. 1,3 biphosphoglysylate. But also this reaction, it is enhanced by this enzyme ambayo inaitwa glyceraldehyde 3 phosphate dehydrogenase. So we are tend to add the phosphate group. So hapo unaweza kuona kuna bi, meaning that there is presence of 2 phosphate group in that molecule. And after formation of the 1,3 biphosphate group, now we will form another thing, that is the 3 phosphoglycerate. And in order for the formation of this phosphoglycerate, now we are using this enzyme ambayo inaitwa phosphoglycerate kinase. So huyu enzyme ndiye ambaye atakatalyze hii formation, kwa maana kwamba we are removing a phosphate group from this molecule. And removing of the phosphate group from this molecule, it results in the production of the ATP and the formation of the 3 phosphate glycerate. So now you can see after the formation of the 3 phosphoglycerate, also this molecule, it will be converted to form 2 phosphoglycerate. And this in order for the formation of 2 phosphoglycerate, we must have the presence of the enzyme ambayo inaitwa phosphoglyceromutase. So atakatalyze hii reaction ili kupata hii phosphoglycerate, meaning hapa kuna kuwa tu na ile just rearrangement. So after that also in order for the formation of the phosphoenolpyruvate, we tend to remove water. Ndio maana tukasema hii reaction ina involve removing of water, and that reaction it will be catalyzed by this enzyme ambayo inaitwa enolase enzyme. And this baada ya formation ya hii ambayo tunaita phosphoenolpyruvate, in order for the formation of a pyruvate ambayo sasa itakuja kuingia kwenye ile Krebs cycle. So hii huyu enzyme atatumia kuconvert hii phosphoenolpyruvate kwenda kupata pyruvate, na huyo enzyme anaitwa pyruvate kinase. And here we are using ATP and there will be the formation of the ADP. So now unaweza ukaona hii ndio glycolysis process ina result into the production of the pyruvate as the end product. And now after that, let's see another thing, that is the fate of the pyruvic acid. What are the fates which are formed after the pyruvic acid? So the pyruvate produced during the glycolysis has 2 possible fates depending on the availability of oxygen in the cell. So hile pyruvic acid tuliyoona kama ni final product ama ni end product ambayo ilikuwa imetengenezwa kwenye process nzima ya glycolysis, tunasema kwamba hile pyruvate inaweza ikaingia kwenye different 2 kinds of stages. And here my student tukasema kwamba it can be probably enter in the availability of oxygen or in unavailability of oxygen. So kama kukiwa na presence of oxygen itaingia kwenye the next stage. Tutaenda kuona ni stage gani ambapo hii pyruvic acid itaingia na kwenda kusababisha kuproduce a lot of ATP or just few number of ATP. So it just depend on the availability of the oxygen in the cell. So, my student, for more clarification on the effects of a pyruvic acid, I would like to invite you into the next part of this session so that we can have a clear understanding what the effects of a pyruvic acid in the process of aerobic respiration and anaerobic respiration. So, thank you for your time and for listening.